Hello everyone. So in today's lecture, we will be studying about what are lipid rafts and what is its significance. So let's begin. So in the previous lecture, we had studied that phospholipids are the major class of lipids that constitute about almost half of the total lipids in the cell membrane. But it is also important to understand that cell membrane is not uniform throughout, because there are certain specialized regions or microdomains where the concentration of these phospholipids, especially the unsaturated phospholipids, are very less, and those regions are highly enriched with sphingolipids and cholesterol, etc. So such specialized microdomains in the cell membrane are called as lipid raft. So this is the structure of cell membrane, where these are the raft domain and these are the non-raft domain. So just by looking at this structure, we can clearly see that this structure looks quite more rigid as compared to the non-raft domain, right? So let us know the reason behind it. So as we know that the lipid rafts, they are highly enriched with sphingolipids, and these sphingolipids, they have long saturated hydrophobic tail. So see, if a lipid contains a saturated hydrophobic tail, then the structure of the lipid molecule is something like this, where we draw two straight fatty acid tails. But if a lipid contains unsaturated hydrophobic tail, then due to the presence of a cis double bond in one fatty acid chain, the structure looks something like this, means there is a kink present in one fatty acid tail. So it looks something like this. So if you compare these two structures, you can clearly see that this structure containing the lipid with saturated hydrophobic tail looks more rigid as compared to this structure, right? So due to the enrichment of sphingolipids in these raft domains are kind of more thick and rigid in nature. So secondly, these lipid rafts are also enriched with cholesterol, and this cholesterol is generally packed within these lipid tails. So here in the structure, you can clearly see that these red color parts are nothing but cholesterol that is filling any void between the lipid molecules. So thereby, it is making this domain, this region, much more ordered and less fluid in nature. And thirdly, these lipid rafts, they are also enriched with certain membrane proteins which are involved in signaling and trafficking related processes. So now the next question that comes to your mind is that what are the significance of these raft domain? So let us know the answer for this question. So, if we talk about the significance of these lipid rafts, they serve as specialized sites for cell signaling, because these regions contain cluster of different receptors like GPCR, T cell receptors, and RTKs, certain signaling proteins and enzymes. So, when any extracellular signaling molecule comes and bind to the receptor, it triggers a strong intracellular signaling cascade. So due to the enrichment of different receptors, signaling proteins, kinases, and enzymes in these raft domains, a cell can very effectively and efficiently communicate with its extracellular environment at these domains. Secondly, these domains also serve as great sites for cargo selection and vesicle formation and budding. So, if anything has to come inside the cell or has to go out of the cell, it is often packed into the vesicles at these domains. So, these domains also serve as specialized sites for vesicle trafficking. And apart from this, these regions also serve as ideal entry point for pathogens. So these domains contains specialized HSPs or heat shock proteins that enhances the stability of these domains. And many pathogens use these HSPs as binding sites to enter inside the cell. Like in case of Japanese encephalitis virus, this HSP70 acts as an integral part in the binding and entry of the virus inside the cell. So that is why researchers are designing certain drugs that can disrupt these raft domains to prevent these raft associated entry of pathogens inside the cell. So this was all about lipid rafts and its importance. So if you have any doubts, you can drop it down in the comment section. I'll be happy to clear that. So see you in the next video.